Introducing Group 5 fluid balance. Number 1, adenosine. So let's look at adenosine, otherwise known as adenosine. Adenosine is added to the system and binds to its A2 receptor. The binding results in the activation of the GS protein transduction pathway. GS protein activates adenylyl cyclase. AC then takes ATP and converts it to cyclic AMP. Cyclic AMP then activates protein kinase A. pKa then activates myosin light chain phosphatase. But MLCP is not interested in the pKa anymore and is actually interested in the myosin P. Myosin P is then unfortunately just myosin. But pKa still has a job to do and activates the potassium channel, which allows more potassium ions to leave the cell. This concludes the GS transduction pathway, which causes relaxation. This causes a dilation of arterioles, which increases the radius of the blood vessels, which decreases resistance and increases flow Q. This can be explained in terms of the Ohm's law. This increased flow resulted in a higher drip rate. The dilation also increases the pressure inside the capillaries. As a result, Phi, the flow through the capillary walls, increases. This can be explained by the Frank-Starling hypothesis. This increase in Phi results in a fatter frog.
And let's move on to ADH, or antidiuretic hormone. ADH binds to its receptor, which activates the GQ protein pathway. GQ alpha activates phospholipase C, or PLC. PLC then causes PIP2 to be converted into DAG and IP3. IP3 then binds to its receptor on the sarcoplasmic reticulum. This causes a release of stored calcium. Calcium then binds in a 4:1 ratio to CAM. CAM then activates the myosin light chain kinase, or MLCK. MLCK in turn then phosphorylates myosin. This induces contraction. Throughout this, DAG activates protein kinase C. PKC enhances calcium transport via voltage-gated L channels and phosphorylates potassium channels, which inhibits release of potassium. These effects work together to inhibit repolarization, enhancing contraction. ADH causes the constriction of arterioles. This causes a decrease in the radius of arterioles. This increases resistance, which decreases flow. Looking at Ohm's law, one can see that these effects will cause a decrease in drip rate. ADH also causes vasoconstriction in the veins. Vasoconstriction of the veins causes less fluid to leave the capillaries. This increases capillary pressure, and this causes an increase in Phi. The increase in Phi causes an increase in weight.
Finally, our last drug is histamine, and it binds to H1 and H2 receptors. When histamine binds to the H1 receptor, it activates the GQ pathway. However, in endothelial cells, the GQ pathway has a different effect. Calmodulin can bind onto endothelial nitric oxide synthase, or eNOS. eNOS then produces nitric oxide. Nitric oxide can diffuse out of endothelial cells and enter smooth muscle cells. Once nitric oxide enters smooth muscle cells, it can activate guanylate cyclase, or GC. GC then creates cyclic GMP from GTP. Cyclic GMP then activates PKG, or protein kinase G. PKG then inhibits calcium L-type channels and the IP3 receptor on the SR. This results in relaxation because of less calcium entry. The effects of smooth muscle relaxation are similar to adenosine, which resulted in increased Phi and Q. As explained earlier, increased Phi and increased Q resulted in an increased drip rate and weight. Another effect of the GQ pathway is the contraction in endothelial cells due to activated motor units. This causes gap junctions to be larger and leads to an increase in K, leading to an increase in Phi. Histamine can also bind onto its H2 receptor, which leads to the GS pathway. Let's review that. And that's the end of our project.